Erwin Schrödinger's book, What is Life?, is a seminal work that posed critical questions about the nature of life. Published in 1944, it bridged physics and biology, inspiring scientists to explore life's principles through physics. What is the molecular recipe book for life? Schrödinger proposed that living organisms have a molecular recipe book, leading to the discovery of DNA. DNA contains instructions for growth, development, functioning, and reproduction of all known organisms. Like a cookbook for cells, DNA directs the creation of proteins and essential molecules. Example: the sequence of nucleotides in DNA determines the sequence of amino acids in proteins, which in turn determines protein function. Schrödinger's book suggested a molecular structure carrying genetic information, termed an aperiodic crystal. This idea inspired James Watson and Francis Crick to discover DNA's double helix structure. Schrödinger's interdisciplinary approach showed that physics principles could elucidate biological processes. Example: the double helix model explained how genetic information is replicated and passed on during cell division. Schrödinger described an aperiodic crystal as a stable, complex structure carrying genetic information. Unlike regular crystals with repeating patterns, an aperiodic crystal has a unique, non-repeating pattern, encoding vast information. This concept helped scientists understand DNA's intricate structure. Example: the non-repeating sequence of base pairs in DNA allows for the storage of complex genetic information. Schrödinger, a physicist, applied his problem-solving skills to biology, akin to a detective solving a mystery in a new setting. His fresh perspective led to groundbreaking ideas influencing molecular biology. Example: Schrödinger's approach helped identify the physical basis of genetic mutations and their role in evolution. Think of genetic information as a mosaic made of unique pieces. Schrödinger suggested it might be stored in a similarly intricate molecular structure. Each piece represents part of the genetic code, forming a complete picture, encoding life's instructions. This idea paved the way for understanding DNA's complex structure. Example: the Human Genome Project mapped the entire human DNA sequence, revealing the mosaic of genetic information. What is Life? Provided a framework guiding molecular biology's development. By suggesting life processes could be explained through physical principles, Schrödinger encouraged the investigation of genetics' molecular basis. His ideas have led to advancements in genetics, biotechnology, and medicine. Example: techniques like CRISPR gene editing are direct results of understanding DNA's molecular structure. Schrödinger's interdisciplinary approach was revolutionary. He showed that the principles of physics could be applied to biological questions, leading to a deeper understanding of life's molecular mechanisms. Example: the discovery of the double helix structure of DNA was a direct result of applying physical principles to biological problems. Schrödinger's ideas also influenced the development of molecular biology as a distinct scientific discipline. His work inspired a generation of scientists to explore the molecular basis of life. Example: the field of molecular biology has grown to include a wide range of techniques and technologies, from gene sequencing to synthetic biology. Today, Schrödinger's legacy lives on in the ongoing research into the molecular mechanisms of life. Scientists continue to build on his ideas using advanced technologies to explore the complexities of genetics and molecular biology. Example: techniques like CRISPR gene editing and next-generation sequencing are direct results of the foundational work laid by Schrödinger and his contemporaries. Schrödinger's What is Life? Remains a seminal work in the history of science. It continues to inspire new generations of scientists to explore the fundamental questions of life through the lens of physics and biology. Example: the book's influence can be seen in the continued advancements in genetics, biotechnology, and medicine. As we continue to unravel the mysteries of life, Schrödinger's legacy serves as a reminder of the power of interdisciplinary thinking. His work has shown that by bridging the gap between physics and biology, we can gain a deeper understanding of the fundamental principles that govern life. Example: the ongoing research into the molecular basis of life is a testament to the enduring impact of Schrödinger's ideas. In conclusion, Schrödinger's What is Life? Has had a profound impact on the field of molecular biology. By applying the principles of physics to biological questions, Schrödinger opened up new avenues of research and inspired generations of scientists to explore the molecular mechanisms of life. Example: the continued advancements in genetics, biotechnology, and medicine are a direct result of Schrödinger's pioneering work. Schrödinger's legacy is a testament to the power of interdisciplinary thinking and the importance of asking fundamental questions about the nature of life. His work continues to inspire and guide scientists in their quest to understand the molecular basis of life. Example: the ongoing research into the molecular mechanisms of life is a testament to the enduring impact of Schrödinger's ideas. Erwin Schrödinger, born in 1887 in Vienna, was a Nobel laureate in physics and a philosopher of science, known for his work in quantum mechanics and wave theory. His book, What is Life?, explored the physical aspects of living cells, proposing the idea of an aperiodic crystal to explain genetic information storage. Schrödinger's What is Life? Presents biological entities as organized systems countering entropy. He proposed aperiodic crystals, which store genetic information in a non-repetitive coded form, akin to how DNA sequences encode genetic instructions. This idea predated the discovery of DNA's double helix, laying groundwork for understanding the physical basis of heredity. Schrödinger's ideas directly influenced the birth of molecular biology. James Watson, co-discoverer of the DNA double helix, credited Schrödinger's work for inspiring his research. What is Life? Laid the groundwork for the DNA revolution, impacting gene replication and protein synthesis studies. Schrödinger's intersection with Claude Shannon's information theory was pivotal, framing genetic material as an information storage system. For example, DNA's double helix structure encodes genetic instructions. His foresight into genetics as biological information processing laid the groundwork for decoding the genetic code, a concept central to modern genetics and bioinformatics. Advances in genomics have validated and expanded Schrödinger's hypotheses. For instance, next-generation sequencing has revealed the intricate architecture of genomes, such as the human genome's 3 billion base pairs, confirming Schrödinger's insights into genetic information. Epigenetics has uncovered complex gene regulation mechanisms that Schrödinger could only hypothesize. This section examines how DNA methylation and histone modifications, such as acetylation, impact gene expression without changing the DNA sequence, enhancing our grasp of heredity and evolutionary processes. For instance, DNA methylation can silence tumor suppressor genes, influencing cancer development. Systems biology represents a paradigm shift towards an integrated understanding of biological phenomena. Erwin Schrödinger envisioned life as a complex system, and today's computational models, like those used in genomics and proteomics, echo his early visions. They strive to predict and manipulate biological outcomes, such as gene expression and metabolic pathways, on a systems-wide scale, really bringing to life the depth of his foresight. Investigating quantum mechanics in biological processes was speculative in Schrödinger's era. Modern quantum biology reveals quantum effects in photosynthesis, where excitons transfer energy efficiently, and in enzyme reactions, where quantum tunneling accelerates biochemical processes. While Schrödinger's ideas were foundational, they faced critiques. For instance, his treatment of genetic information as a code was oversimplified. Later discoveries like the double helix structure of DNA by Watson and Crick refined his concepts and addressed these limitations. The field of synthetic biology, where scientists design and create new biological parts and systems, includes creating synthetic genomes and reprogramming cells. This mirrors Schrödinger's vision of mastering the molecular fabric of life. We explore the potential and perils, such as bioengineered organisms for medicine and environmental solutions. This field continues to push the boundaries of what we understand as life. Reflecting on Schrödinger's legacy, His ideas on molecular biology have led to the discovery of DNA's double helix structure. What is life? Inspired interdisciplinary research such as biophysics and systems biology, enhancing our understanding of genetic coding and cellular processes. It continues to guide us in exploring quantum biology and other emerging fields.